Okay, so let's dive into something really uh, fundamentally puzzling about our genes.
Hmm.
Imagine this. Two different people, nearly identical genetic makeup, right? And both are missing the exact same tiny piece of chromosome 15. Yet one person gets Prader-Willi syndrome, think initial floppiness, then this uncontrollable hunger later on, and the other gets Angelman syndrome, severe developmental delays, no speech, but often this really happy, excitable nature. How does that even happen? How can it matter if that missing bit came from mom versus dad?
It's a fantastic question, and honestly, it's one of the most elegant paradoxes in biology. It tells you straight away that genetics isn't just about the sequence, the A's, T's, C's, and G's. What we see, the phenotype, depends hugely on this whole other layer of control, how the DNA is packaged, how it's regulated. So today we're going deep into epigenetics, specifically this phenomenon called genomic imprinting. Our mission really is to explore how genes get expressed differently based purely on which parent you inherit them from and why this happens at all.
Right, and it all starts with that basic, almost physical challenge you mentioned earlier, cramming 2 meters of DNA into a cell nucleus that's just incredibly tiny, like 10 micrometers across.
Exactly. It's an insane packaging feat. Think about fitting miles of thread into a tiny bead. But crucially, this packaging isn't just storage, it is regulation. Tightly packed DNA, generally off. Loosely packed, generally on.
So the cell needs a way to manage this packing, to keep things stable but also flexible. This is where chromatin comes in, right? DNA wrapped around histone proteins.
Precisely. And you need stable patterns, so a liver cell stays a liver cell generation after generation. But you also need to turn genes on and off dynamically. You need that switch between open, active states, that's euchromatin, and tightly packed, silent states, heterochromatin. And controlling that switch, that's the job of the epigenetic toolkit.
Okay, let's unpack that toolkit. What are the main players here that give cells their stable identities?
Well, there are three core mechanisms people usually talk about. DNA methylation, histone modifications, and also non-coding RNAs play a role. But for our story today, for genomic imprinting, the absolute star is DNA methylation.
Methylation, so adding a chemical tag directly onto the DNA.
Exactly. It's a tiny molecule, a methyl group CH3, that gets added onto a cytosine base, usually when it's next to a guanine. We call that a CpG site. And these CpG sites are often found bunched together in really important places, like gene promoters or these special regions called imprinting centers.
Okay, so you stick this methyl tag on. What does it do? Does it damage the gene?
No, not at all. It doesn't change the sequence. Think of it more like
Hmm.
putting a chemical off switch on the gene. It can physically block the machinery that reads the gene, or it can recruit proteins that basically lock down that whole region of DNA. And the key thing is this mark is copied when the cell divides. There's an enzyme, DNMT1, that makes sure the pattern is maintained on the new DNA strand. It creates the stable epigenetic memory.
Got it. So methylation provides the long-term memory on the DNA itself. But you also mentioned histone modifications. That sounds more about the packaging structure, or is called the histone code.
Yeah, the histone code is fascinating. Histones have these flexible tails sticking out, and they can be decorated with all sorts of chemical tags, acetylation, methylation, phosphorylation. Think of these tags like little signals or flags on the chromatin structure.
Traffic lights, maybe. Green means go, red means stop.
That's a great analogy, absolutely. For example, adding certain methyl groups to histone 3, like at position K4, H3K4 methylation, that's usually a go signal. It marks active genes, loose chromatin. But methylating a different spot, like H3K9, is often a strong stop signal. It recruits silencing machinery, helps form that dense, inaccessible heterochromatin. The specific combination of these marks, the code, tells the cell's machinery where to open things up and where to keep them shut tight.
Okay, so we had these tools. Methylation for the stable memory, histones for the dynamic structure. Now, let's bring it to genomic imprinting, this parent of origin thing. Why on earth would evolution decide to silence a perfectly good gene copy just because it came from dad or from mom?
The why. That's still debated, but a leading idea is the parental conflict hypothesis. It's kind of an evolutionary tug of war. The theory suggests paternal genes might favor maximizing fetal growth, extracting more resources from the mother.
Right, to ensure the offspring survives, maybe at the mother's expense.
Sort of, yeah. Whereas maternal genes might favor conserving resources, limiting growth slightly to ensure the mother can survive and have more offspring later. It sounds a bit dramatic. But this conflict could have driven the evolution of imprinting for certain growth-related genes.
That's pretty wild. So The outcome is that for these specific imprinted genes, and it's not most genes, right? Only a small fraction.
A very small fraction, yeah. Maybe around 165 or so known human genes are imprinted. Over 99% are expressed from both copies. But for these imprinted genes, the result is monoallelic expression. Only one parental copy is active, the other is silenced by these epigenetic marks. And just to be clear, paternally imprinted means the copy from the father is silenced, so the mother's copy is active. Maternally imprinted means the mother's copy is silenced, so the father's copy is active.
Okay, and the really mind-bending part is how these imprints are handled across generations.
Yeah.
You called it the imprinting cycle.
Yes, it's quite remarkable. So, stage one. The specific methylation marks, the imprints, are established in the parent's sperm or eggs, depending on their sex. A father puts paternal marks on his sperm. A mother puts maternal marks on her eggs. Stage two. These marks are passed down to the embryo and are generally maintained in all the body's somatic cells throughout life. They help keep cell identity stable.
But then, the reset button.
Exactly. Stage three. When that new individual grows up and makes their own sperm or eggs in their germline cells, all the old inherited imprints get completely erased, wiped clean. And then new imprints are established based on that individual's sex. So a woman erases the imprints she got from her dad and puts down only maternal marks on all her eggs. A man erases his mother's imprints and puts down paternal marks on his sperm.
Wow, that erasure and resetting seems, incredibly precise, but also risky. You have to get it exactly right.
It is high stakes. Errors in this resetting process can actually cause imprinting disorders.
But it perfectly explains our starting puzzle, the Prader-Willi-Engelmann paradox. If you have a deletion on chromosome 15, the effect depends entirely on whether you lost the copy you got from your dad or the one from your mom, because only one of them was meant to be active in that region anyway.
Precisely. You delete the active copy, you get disease. You delete the silent copy, you might be perfectly fine, a carrier perhaps, but phenotypically normal.
Okay, let's ground this in the case study. That critical region, 15q, 11q13. First, Prader-Willi syndrome, PWS. You describe the clinical picture. The floppy baby stage, poor feeding early on, and that switch to intense hunger, obesity risk, some learning difficulties.
Right. PWS results from losing the function of genes in this region that are normally expressed only from the paternal chromosome. The maternal copies are silenced by imprinting. Key genes involved include SNRPN, MAGEL2, and a cluster of small RNAs called SNORARD116. Lose the paternal copies, you lose their function, particularly affecting the hypothalamus, which controls hunger and satiety.
In most cases, like 70–75%, are a straightforward deletion on the dad's chromosome 15. But then there's this other cause, maternal uniparental disomy, UPD. That accounts for maybe 20–30%. How does someone end up with 2 copies of mom's chromosome 15 and none from dad?
That's a fascinating mechanism, usually involving what's called trisomy rescue. Sometimes very early in development, an embryo might accidentally start with 3 copies of chromosome 15, 2 from mom, 1 from dad, or vice versa. That's usually lethal. To survive, the embryo tries to correct this by randomly kicking out one of the extra chromosomes. If it happens to kick out the paternal chromosome 15, it's left with 2 maternal copies. And since the PWS genes on the maternal chromosome 15 are normally silenced by imprinting anyway, having 2 silenced copies gives you the exact same functional outcome as deleting the active paternal copy, no active PWS genes.
Okay, that makes sense. It's a rescue that still leads to the syndrome. Now, the flip side. What if the maternal contribution to that same 15q11q13 region is lost instead?
Then you get the completely different picture of Angelman syndrome, AS. Severe developmental delay, often no speech at all, problems with movement and balance, ataxia seizures are common, microcephaly, and that characteristic happy demeanor with frequent laughter.
So what's the key maternal gene here that's lost?
The critical gene for AS is UBE3A. In the brain, particularly in neurons, UBE3A is primarily expressed from the maternal chromosome. The paternal copy of UBE3A is largely silenced in neurons due to imprinting. So if you lose the maternal copy of UBE3A through deletion, or if you have paternal UPD for chromosome 15, meaning 2 paternal copies, both with silenced UBE3A, or a mutation in the maternal UBA3A gene itself, or an imprinting defect that silences the maternal copy, the end result is loss of UB3A function in the brain.
And UB3A, what does it normally do in the brain? Why does losing it cause these specific symptoms?
UB3A is an enzyme called an E3 ubiquitin ligase. Its job is to tag other proteins with ubiquitin, which often marks them for degradation, basically, cellular recycling or disposal. This protein turnover process is absolutely crucial for normal brain development and function, especially at the synapse, the connection points between neurons. It's involved in synaptic plasticity, learning, and memory. So when you lose UB3A function in neurons, you disrupt this delicate balance of protein turnover, which leads to the profound neurological problems we see in Angelman syndrome.
So to summarize the core connection, PWS and AS, same chromosomal region, 15q11q13, totally different disorders. PWS is loss of paternally expressed genes in that region. AS is primarily loss of the maternally expressed UBE3A gene in that region.
Yeah.
It really just comes down to which parent's on switch got broken.
Exactly right. It's all about the parent of origin and which copy was epigenetically programmed to be active.
Which brings us to diagnostics. If the cause can be a deletion, or having two copies from one parent, UPD, or even just the wrong epigenetic label on perfectly normal DNA sequences an imprinting defect, then just sequencing the DNA isn't enough, is it?
No, it's not sufficient on its own. You absolutely need to assess the epigenetic state, specifically the methylation pattern in that critical region.
And the go-to spot for that is the SNRPN gene's promoter region, right? Because it has that clear parent-specific methylation pattern.
That's the key diagnostic target. Within the SNRPN locus, there's a region, often called the imprinting center, where the maternal allele is heavily methylated, covered in those methyl tags. The paternal allele, in contrast, is normally unmethylated. It's naked DNA in that specific spot. This difference is consistent and reliable.
So the test is basically asking, do we see both patterns or just one?
Precisely. A normal individual will show both the methylated maternal pattern and the unmethylated paternal pattern. If the test only detects the methylated maternal pattern, it implies the unmethylated paternal contribution is missing. That points strongly towards PWS, either by deletion or maternal UPD. Conversely, if the test only detects the unmethylated paternal pattern, it means the methylated maternal contribution is absent. That indicates AS, usually due to deletion or paternal UPD.
How do they actually measure this methylation? I know older methods use restriction enzymes.
Yeah, the classic method involved methylation-sensitive restriction enzymes. These enzymes cut DNA only if their recognition site is unmethylated. So you could digest the DNA, run it on a gel, and see different band patterns based on what got cut. Normal would show two bands. PWS, lacking the unmethylated paternal copy, would only show the larger band from the uncut maternal allele. AS, lacking the methylated maternal copy, would show only the smaller band from the cut paternal allele.
But now there are faster ways, like methylation-specific PCR, MSPCR.
Yes, MSPCR is much more common now. It relies on a really clever chemical treatment first, bisulfite conversion. What bisulfite does is it chemically changes unmethylated cytosines into uracil, which behaves like thymine in PCR. Critically, it leaves methylated cytosines untouched.
Ah, so you chemically alter the DNA sequence based on its methylation status?
Exactly. Now you have sequence differences between the originally methylated and unmethylated strands. Then you design two pairs of PCR primers. One pair will only bind and amplify the sequence that came from the methylated maternal DNA after bisulfite treatment. The other pair only amplifies the sequence from the unmethylated paternal DNA after treatment. Run the PCR products out. Normal gives you two bands, maternal and paternal products. PWS gives you only the maternal band. EDS gives you only the paternal band. It's very elegant and quite definitive.
That really is elegant. Okay, so we've gone from this huge DNA packaging problem through the epigenetic tools like methylation and histone codes to this specific subtle regulation called genomic imprinting. And we saw how disrupting it just based on whether a gene came from mom or dad causes devastating but distinct syndromes like PWS and AS. It hammers home that genetics isn't just about typos in the code, but about how the instructions are written and read.
Absolutely. And the fact that imprinting disorders highlight problems with the regulation, not necessarily the gene sequence itself, opens up some really interesting therapeutic thinking. Epigenetic marks, unlike DNA sequence mutations, are potentially reversible. They can respond to environmental cues, to drugs. So it raises this profound question for the future. Instead of focusing solely on fixing the DNA sequence, could we develop ways to safely rewrite or correct these faulty epigenetic instruction labels? Could we, for example, reactivate the silent, healthy paternal UBE3A copy in neurons of individuals with Angelman syndrome, or turn on the silent maternal PWS genes? It's a major challenge, but targeting the epigenome, correcting the label rather than the code, could be a whole new frontier for treating these kinds of genetic disorders.